right, let's talk about one of the most infamous viruses out there, rabies. Welcome to part 1 of our deep dive. We're going to cover everything you absolutely need to know for your exams, from what this thing looks like all the way to how it wreaks havoc on the body.
Okay, so here's our game plan. We're going to start with the enemy itself, its morphology. Then we'll look at the different strains you'll encounter. After that, we'll trace its path of invasion, the pathogenesis, and we'll wrap up with the aftermath, the clinical phases.
All right, first things first, Section 1, the enemy. Let's get up close and personal with the rabies virus and its structure.
So, if there's one thing you tattoo on your brain about this virus, it's this. It's bullet-shaped. That is a classic exam question right there. It's an enveloped, single-stranded, negative-sense RNA virus from the Rhabdovirus family. But honestly, that bullet shape is your biggest clue. You see bullet-shaped virus, you think rabies. Period.
And here's what we're talking about. Look at this. On the left, you've got the schematic, right? You can see those glycoprotein G spikes sticking out. Super important, we'll get back to those. And inside, you've got the nucleocapsid. But then, look at the right. That's the real deal, an electron micrograph. You can't miss that classic bullet shape. It's literally a bullet.
Okay, now this table is super high yield, so listen up. There are two main antigens you gotta know. First, glycoprotein G. Think of this as the virus's skeleton key. It's on those outer spikes, and it's what lets the virus into our nerve cells. Because it's on the outside, it's also what our immune system sees and makes protective antibodies against. Make sense? Right? Then you have the nucleoprotein. This one's on the inside, so antibodies against it, not protective. But, and this is a big but, it's what we look for when we're diagnosing rabies. So G for get in and good for immunity, N for not protective, but needed for noticing the virus in the lab. We'll hit diagnostics hard in part 2.
Okay, moving on. Section 2, the strains. You've got street virus and fixed virus, and you bet this is a favorite comparison question on exams. All right, let's break this down. Street virus is exactly what it sounds like. It's the wild one, what you'd get from an animal bite in the street. Its incubation period is long and all over the place. And, the big one, it produces Negri bodies. Now, fixed virus is the lab rat. We've passed it through animal brains over and over until its incubation period is short and fixed. Get it? Like 4 to 6 days. Super predictable. It's great for making vaccines. But the absolute killer difference you need to remember? Fixed virus does not produce Negri bodies.
And because we love a good mnemonic, here you go. Just remember FIX. The I is for inclusion bodies. That's our Negri bodies. And the X, well, that's just to remind you they are absent, crossed out, non-existent in the fixed virus. FIX. It's simple, it works, use it.
Okay, now for the main event. Section 3, the invasion. This is the journey of the virus from bite to brain. This pathway is everything for understanding rabies.
All right, take a good look at this diagram, because this is the whole story in one picture. We're going to follow the virus on its entire horrifying road trip, step by step. So here's how it goes down. Step 1, the bite. The virus is in. Step 2, it doesn't just sit there, it starts multiplying right there in the muscle tissue. Step 3 is the big one. This is where it makes its move. It jumps onto our own acetylcholine receptors at the neuromuscular junction and gets into the peripheral nerves. It's like it's picking the lock to our nervous system. From there, it's an express train to the brain. This is called centripetal spread, traveling towards the center. It hitches a ride on our nerves, moving backward. That's retrograde axonal transport all the way up to the spinal cord. And once it's in the spinal cord, it's a straight shot to the brain. But it doesn't stop there. Once it's taken over the brain, it turns around and spreads back out, that's centrifugal spread, to all sorts of organs, but most importantly to the salivary glands. And why? So it can get into the saliva and be ready to infect the next victim. It's a terrifyingly smart cycle.
So when the virus is multiplying in the brain, what's the evidence it leaves behind? The classic calling card is the Negri body. When a pathologist looks at an infected brain slice, this is the smoking gun they're searching for. They're these little packets of viral proteins, these eosinophilic inclusions, right inside the cytoplasm of the neurons.
And here's what they look like. See those little pink dots inside the neuron? Those are Negri bodies. Now, finding these is pathognomonic. That's a fancy word meaning if you see them, it's 100% rabies. But, and this is a huge clinical pearl, not seeing them doesn't rule it out. About 20% of cases might not show them. And remember our mnemonic, FIX? You only see these with the wild street virus, never the fixed lab strain.
Okay, so the virus has made its journey. Now what? Section 4, the aftermath. What does this look like for the person who's been infected? Let's talk about the clinical phases.
This timeline is, well, it's terrifying. Look at that incubation period, 20 to 90 days, but it can be super variable. And here's a key point for your exams. The length of this period depends on the bite location. A bite on the foot, longer incubation. A bite on the face, right next to the brain, much, much shorter. But once you leave the incubation period and the first symptoms of the prodromal phase start, things happen fast. From there, it's just days until the acute neurologic phase and then coma and death. It is a relentless and rapid decline.
Okay, when we get to that acute neurologic phase, rabies can present in two different ways. The one everyone thinks of, about 80% of cases, is furious rabies. This is the classic picture: agitation, hyperexcitability, and those hallmark signs, hydrophobia and aerophobia. Hydrophobia, fear of water, isn't really a fear, it's that any attempt to swallow causes these incredibly painful spasms in the throat. Same with aerophobia, the fear of air from a draft, it's just awful. The other 20% of cases are paralytic or dumb rabies. This one is less traumatic, which can actually make it harder to diagnose. It starts with paralysis in the limb that was bitten and just spreads. It's a flaccid paralysis that moves through the body.
So, the virus's journey is brutally efficient, and when symptoms show up, it's almost always fatal. This leaves us with a huge question. How do we fight back? How can we possibly detect this thing early enough to make a difference? And most importantly, how do we stop this whole process from even starting after a bite? That's what we're going to cover in Part 2. We'll get into the lab diagnosis, treatment, and the absolutely crucial topic of post-exposure prophylaxis. I'll see you there.